Right. So I'm going to be talking to you today about a couple of different mRNA therapeutics. To mention my conflicts, we've got a bunch of patents, and I'm on the SABs of a few companies. Uh, so let me start off with describing the background for mRNA. And the first time RNA was injected into an animal with the idea of using it as a therapeutic protein delivery system was in 1990. RNA therapeutics went nowhere after that, but vaccine studies proceeded, and these mostly involved taking dendritic cells, which are antigen-presenting cells, out of a mouse and then out of a person, pulsing them with RNA and giving them back. The reason why RNA therapeutics didn't develop was because of this. There are 17 different innate immune sensors that recognize RNA and produce a variety of effector functions. They make pro-inflammatory cytokines, type 1 interferons, they directly block translation, and they induce apoptosis and cell death. So the work that Katie and I did was to understand the immunity of RNA. And this was one of our telling experiments where we took RNA derived from a variety of cell sources of bacteria and mammalian cells and tested them for their ability to induce inflammatory cytokines. And what was striking was that tRNA was non-immunogenic, while mitochondrial and bacterial RNA was highly immunogenic. So that gave us a hypothesis that maybe it's nucleoside modification. tRNAs up to 25% of the nucleotides are modified. Bacteria, very few. For modifications, there's over 100 different types. They include isomerizations, methylations of the base and of the sugar. They occur at conserved positions, and it's recently been found that M6A is reversible. So to test this, we made RNA that contained modified nucleosides. So here, every C has a 5-methyl group. Every U is a pseudouridine. And we tested these RNAs for immunogenicity, and many modifications reduced the immunogenicity. But what was striking is when we tested, could those modified RNAs be translated? Not only were they translated, but they were translated better, up in this case in culture, 10-fold better than an unmodified mRNA. That was unexpected. We also found that there were contaminants when RNA is made, and these are double-stranded. And if we remove those contaminants, we further increased translation and got rid of all the innate immune activation. But strikingly, what that did is it increased the protein production from the RNA by about 4 logs, so a huge increase in amount of protein produced. So this is now 2005. Some of you may not have been born yet, but we were asked the question, why? Who cares about RNA? And our response was that protein therapeutics is a huge part of the pharmaceutical industry, and it continues to grow. The way those proteins are made are in 50,000-liter drums full of CHO cells or another cell that secrete the protein you want. You then have to invent a purification procedure to get that protein away from cell culture contaminants. Every protein, you have to reinvent both of these. With mRNA, it's a single vessel makes the RNA, a single reaction purifies it, and those reactions are identical no matter what the coding sequence is. So our thought was that RNA would be safer, it would be a simple GMP production with a reduced cost and increased potency. So I'm going to mention just a little bit of vaccines because you guys aren't immunologists and I don't want to bore you too much. But I wanted to talk about this vaccine because it has huge importance going forward. So hopefully all of you have had your COVID-19 vaccine, but there have been 3 coronavirus epidemics or pandemics in the past 20 years. What you have to assume is that there's going to be more. So what we were interested in developing is a vaccine that would protect against any beta coronavirus that can cross over from a bat into a human, and also expand this to address all of the variants that are appearing. So the next time there's a coronavirus outbreak, the vaccine will be ready to go instead of having to wait a year to make it, and who knows how long for people to take it. So we've made 6 different immunogens. This is one that was recently published. And what we did is we took the RBD region, the receptor binding domain of the spike protein, and attached it to ferritin. Ferritin forms a 24-mer nanoparticle. And with the RBD attached, you end up with a nanoparticle expressing the RBD domain. Immunologically, nanoparticles are more immunogenic. So these were put into mice and into macaques. And then the sera was analyzed for neutralization of a diverse series of beta coronaviruses. So SARS-CoV-2 is what's going around right now. SARS-CoV-1 is the 2002 SARS outbreak that rapidly disappeared. These are 2 bat coronaviruses that have the potential to cross over into humans. They use the same receptor. They're somewhat closely related to the SARS-CoV-2. And what we see is that this immunogen very potently neutralizes all of these coronaviruses, including distant bat. We see the same results in macaques. And we expanded the analysis to include pangolin and a couple of other bat coronaviruses. But we found that MERS, so COVID-19 is a 2B, MERS is a 2C, MERS was not addressed. The mechanism of how this vaccine works is shown here. So this is a cryo-EM of the spike protein with 2 different antibodies bound. 1041 is a monoclonal that looks like the typical response you see in vaccinated and infected people. It binds to the top of the RBD. But this vaccine induced a different kind of response that's characterized here by 1047. And 1047 binds the side of the receptor binding domain. You don't find 1047-like antibodies in natural infection, and you find them at low levels with the RNA vaccines. What's critical about this 1047 epitope is that it recognizes a highly conserved region of the receptor binding domain. So these are, blue is completely conserved, and the green is slight modifications. We believe this is the reason why this vaccine induces a response that protects against distant beta coronaviruses and bat coronaviruses that might cross over someday. So our conclusion is we've developed 6 new immunogens, I'm showing one here, that are based on conserved epitopes in beta coronaviruses and are able to protect against a broad variety of coronaviruses. We're currently developing MERS 2C immunogens, and we'll hopefully test these in people in 1.5 to 2 years. So what I wanted to talk about, and what's likely to more interest you guys, are studies of targeting lipid nanoparticles. So you all know how lipid nanoparticles are made. They bind ApoE, they target the liver, they target dendritic cells and a few other cell types that have ApoE receptors. But what happens if you want to target a different cell type or a tissue? So we've been working on a number of different targeting approaches, and we've targeted endothelial cells, T cells, and bone marrow stem cells. And I'll show you the data from these. So the way we've initially targeted LNPs is we took a PEG lipid with a longer carbon tail and attached the antibody or scFv or receptor ligand or any targeting molecule to the end of a PEG molecule and inserted it into a lipid nanoparticle. Now that you know the PEG lipids that are used to make LNP rapidly fall off, but because these have an extended carbon tail, they remain with the lipid nanoparticle. When we examine them, the addition of the antibody didn't change the morphology. It increased the size a little bit. The surface charge remained the same, but most importantly, they retain function. So these are cells that don't express PCAM. The LNPs have an anti-PCAM antibody added to them, and they bind very effectively to PCAM expressing cells. But importantly, when they bind, they're taken up and they express the mRNA in the LNP. So the LNPs retain function. These are human UVECs that express PCAM, and you can only see expression of the mRNA when the LNPs are targeted to bind PCAM. We injected these PCAM antibody labeled LNPs into mice, and we saw two things. The first is we saw a reduction in liver uptake. So this is an IG control or a PCAM labeled LNP. There's a significant drop in luciferase expression. And importantly, in the PCAM targeted mice, the lungs lit up. The lungs have the highest density of endothelial cells that express PCAM. We looked at CNS targeting as another direction. And I'll briefly show you these data. The model system is we take mice and we inject TNF alpha, a pro-inflammatory cytokine, into one side of the brain. That leads to local inflammation. In this case, we targeted the LNPs with VCAM. And what you can see here, the uptake in the mice is similar. But when we look at the brain, the VCAM LNPs are binding to the side of the brain that received TNF, that's inflamed. And there's minimal expression on the contralateral side. So we're getting selective uptake of VCAM targeted LNPs to the site of brain inflammation. We wanted to test, could we develop a therapeutic using nucleoside modified mRNA? So these are the control experiments. And what we see is that when we inject TNF alpha, we see inflammation is manifested by vascular leakage. So radiolabeled albumin is leaking out of the blood vessels on the TNF treated mice on the same side of the brain, but is not leaking out very much on the opposite side of the brain. We delivered a VCAM targeted LNP that had a thrombomodulin mRNA. Thrombomodulin is an anti-inflammatory, anticoagulant protein that cells produce to protect themselves. And what you can see is that the animals that receive the thrombomodulin, their amount of vascular leakage goes back to control levels. So we completely blocked vascular leakage induced by inflammation on that side of the brain. And we're continuing to develop other therapeutics to move this further. I'm an immunologist, so I, of course, have an interest in T cells, and we've developed a couple of different T cell targetings. The reasons that we did this is there are many therapeutics that rely on modification or changing T cells. So the classic are CAR T cells, which are targeted cancer therapy that are incredibly effective for a couple of liquid leukemia tumors. But there are many other therapeutics. HIV is resident in resting CD4 positive T cells. So if you can target the T cells and deliver an anti-HIV agent, you may be able to cure HIV. So the key is now, how do you target T cells in vivo with a lipid nanoparticle? And to do this, we initially took an anti-CD4 antibody and linked it to the surface of a lipid nanoparticle. And we saw the same thing we saw with PCAM. There was a significant reduction in liver targeting and a huge increase in spleen targeting. Spleens have high levels of CD4 positive T cells. But there's a problem, and that's T cells do not endocytose. They don't have free endocytic activity where they can take up free particles. Our idea was that if we used an antibody to target the T cells, that endocytosis after binding, it would bring the LNP into the cell and allow the RNA to escape and be translated. Here we're looking at purified human CD4 positive T cells, and we see that when they're targeted against CD4, we have high levels of RNA expression. So we were lucky. We did biodistribution studies, and what you can see from the in vivo injection is that the spleen lights up when we target CD4, but we did something else. And we took the animals after we removed all of these organs and we imaged them again. And what we see is that on the animals that received the CD4 targeted LNPs with luciferase mRNA, we're getting signaling showing up in para-aortic and inguinal lymph nodes. So this means that the LNPs are escaping the circulation, traveling to the lymphatic vessels, traveling to lymph nodes, targeting CD4 positive cells, and being taken up and expressed the mRNA. Here we purified the cells from the spleen, and we purified the T cells, and we showed that T cells are in fact expressing the mRNA. We then moved on to a Cre-Lox system to identify targeted cells, and we delivered Cre recombinase encoding mRNA. So the Cre recombinase removes a stop codon flanked by LOXP sites in front of a fluorescent protein. So when Cre is delivered and is effective, the cells turn green. We analyzed levels of gene recombination after a single injection of anti-CD4 targeted. And in our best experiments, up to 80% of the spleen CD4 positive T cells expressed the green protein, showing they had taken up the LNPs and expressed the RNA. We also looked at lymph nodes, and again, we saw high levels of gene recombination in the lymph nodes, showing that in fact that picture earlier was correct. We then moved on and studied a different T cell marker. So CD4, of course, only targets CD4 positive T cells, macrophages, dendritic cells, and a few other cell types that may be able to express CD4. We wanted a pan T cell marker. So we looked at a bunch and we identified CD5, which is a scavenger receptor. And we chose this one because scavenger receptors endocytose after binding. So we developed CD5 targeted LNPs. And again, we saw very high levels of gene recombination, 80% in the spleens of these animals, high levels in the lymph nodes as well. And then we wanted to do a therapeutic. So the therapeutic we chose was delivering CARs. CARs are molecules that have a T cell signaling domain linked to an antigen recognition domain. These are used for the treatment of a few different leukemias. But what's done is a patient is leukapheresed. They put them on a machine that pulls out 2 billion T cells, 2 billion blood cells from the patient. Those cells are then taken and stimulated to expand the T cells. After 10 days, those cells are infected with a lentivirus that delivers the CAR gene and allows those cells to express the CAR T molecule. They're then given back to the patient. It's a very effective therapy. It costs about $500,000 a dose because of all of that extensive cell culturing ex vivo. What we wanted to know is, could we deliver CAR molecules in vivo? So now you've got an off-the-shelf therapy where you thaw the LNPs and inject them in and you're done. The model we used is a model of cardiac fibrosis. So this was developed by John Epstein and Haig Agajanian. And what they did is they first started off studying human heart. And the red are the cardiomyocytes. The green stain is fibrosis. And what they found is that in both hypertrophic and dilated cardiomyopathy, as well as many other cardiomyopathies, there's an increase in fibrosis. They looked at a gene signatures and they found that the fat molecule had the highest level of differential expression between the cardiomyopathies and the normal heart. They set up a model in mice where they made the mice hypertensive with angiotensin II and phenylephrine. And what they found is that after a week of hypertensiveness in the mouse, fibrosis began to appear throughout the myocardium. So the idea was that we would deliver a CD5 targeted LNP expressing the CAR molecule that gets taken up, the RNA is released, the CAR is translated and put on the cell surface, and then it could attack FAP-expressing fibroblasts and reduce the amount of fibrosis in the heart. Our initial studies, we wanted to make sure we got expression. So these are T cells that were transfected with targeted LNPs in vitro. 80% of the cells expressed the FAP CAR. These animals were injected in vivo. We took out the spleens after 48 hours. 20% of the cells expressed the FAP CAR. We treated hypertensive or control mice with the mRNA LNPs, a single injection. And this is cardiology talk here. All of these are measures of cardiac contractivity, cardiac function. What's important to look at is ejection fraction. So that's the amount of blood that the heart can expel. In fibrotic conditions, the ejection fraction is reduced. When they were treated with the CD5 targeted FAP CAR mRNA, it returned to baseline. And all of these other measures show the same thing, that treatment with the FAP CAR targeted LNPs cured the fibrosis. So what we've demonstrated is we can target T cells in vivo, deliver a CAR molecule, and treat a disease. This is being expanded to treat a variety of fibrotic diseases and other things such as autoimmunity, HIV, other things. The last thing I want to talk to you about is bone marrow stem cell targeting. So again, as an immunologist, this is critically important to me because in my younger days, I used to treat immune deficiencies. All immune deficiencies are bone marrow stem cell defects. Now, the issue with stem cell targeting, and then what I want you to look at is down here. When you take bone marrow out of a human or out of a mouse, and you analyze 700,000 cells, 14,000 of them are CD34, which is a marker of stem cells and progenitor cells. Less than 1,000 of those cells are true repopulating stem cells. So there's a huge loss or huge lack of stem cells in the bone marrow, which makes targeting them very difficult. We worked with Hans-Peter Kim, who identified a marker of human and macaque repopulating bone marrow stem cells called CD90. We attached CD90 to the LNPs and then put a GFP mRNA in them and treated bone marrow from a human in culture. And we're now staining this bone marrow for CD34, and we're looking for the percent of CD90 cells that have taken up the LNPs. And the frequency is about equal to what the frequency of CD90 cells is in the bone marrow. So we had close to 100% targeting in vitro. We then moved to a humanized mouse. So humanized mice are immune deficient mice that have a human immune system along with thymus and liver put in. So they create human immune systems, which include human stem cells that repopulate the mouse. We injected the CD90 targeted LNPs into these animals. We saw no drop in CD34 expression, which was good. We took the bone marrow out and we did an assay that measures the ability of bone marrow stem cells to turn into progenitor cells and produce daughter cells. So the same thing they do in us every day. We're measuring their ability to form progenitors in different colonies. These are 5 different colonies of cells that are produced from bone marrow stem cells. Importantly, what we found is that CD90 binding by the LNPs did not change their efficacy, their ability to form daughter cells and daughter colonies. Also of interest, when we analyzed the animals for CD90 expression, we found none after the CD90 LNP treatment, likely because the CD90 was downregulated and we were no longer able to bind it. We then took these animals and we analyzed them for luciferase expression. These are Ig control LNPs, and the majority of expression is in the liver with some in the spleen. We also looked at CD90 targeted LNPs. We've blocked out the liver, so you're not seeing that. But what you do see is that at a very high resolution, the bones are lighting up with luciferase activity. This is luciferase that has to, the light has to show through the bones. At a much lower resolution, we can see something interesting. You'll see that there's little dots of bright luciferase activity. Those are sites where the bone has been cut. And what these are, they're luciferase activity coming out of the end of the cut bone, showing that in fact the bone marrow has taken up the mRNA LNPs and is expressing the mRNA. We moved on to mouse targeting because we had more flexibility in the kind of therapies we could do. We looked at a number of different bone marrow stem cell markers, and we chose CD117. CD117 is expressed by both bone marrow stem cells and progenitor cells. So it isn't as specific as CD90. When we looked at whole bone marrow, we saw high levels of CD117 directed uptake and high levels of background. This is whole bone marrow that's full of macrophages and dendritic cells that take up LNPs, whether they're targeted or not. We then did an analysis where we looked at what are called the lineage negative cells. So here we're ignoring all of the macrophages, dendritic cells, NK cells, other cells that could be taken up. These are mainly progenitor and bone marrow stem cells. And what we saw is a very potent dose-related increase in luciferase expression with CD117 targeting. We then went back to our AI6 mice. So what we've done here is we've injected either control IG LNPs or anti-CD117 targeted LNPs. And then we let the animals sit for a week and we examine bone marrow stem cells in these animals. This is the percent of bone marrow stem cells that were gene recombined, that the Cre recombinase caused expression of the green fluorescent protein. And we can see that 80 to 90% of the cells took up the LNPs and recombined. We've been following these mice for 6 months. So here, this includes progenitor cells. At 6 months, we're only measuring repopulating bone marrow stem cells. And the levels in these animals are still in the 60 to 70% range. So that's enormous levels of bone marrow stem cell targeting in efficient gene recombination with a single injection of mRNA LNPs targeted to bone marrow stem cells. So there's a bunch of conclusions from this. And when you're thinking about distant therapeutics, number one on our list is sickle cell anemia. So there's currently an FDA-approved protocol to treat sickle cell gene therapy. And that involves taking bone marrow out of a patient, infecting it with a lentivirus that delivers a gene that fixes the beta hemoglobin and giving the bone marrow back. It works, it's effective. The problem is 200,000 people a year are born with sickle cell in Africa. You can't do 200,000 bone marrow biopsies and viral infections. It's the infrastructure, the work, and the cost are excessive. What we're developing is a single IV injection that will target the bone marrow stem cells and will put RNA encoding machinery that can either fix the beta hemoglobin gene or insert a fetal hemoglobin gene or a variety of other cures. This then has expansion to the many bone marrow genetic disorders of red cells, granulocytes, lymphocytes, all bone marrow cells, and further development to target other stem cell genetic defects. So I need to thank all of the people who did these studies, the people in my lab that were involved, Acuitas Therapeutics, who did all of the LNP formations for us, John Epstein's lab, Hans-Peter Kim's lab, Vlad's lab helped us with designing the targeting molecules. And I'll stop there and take questions.
Thank you very much, Dr. Weissman. Let's open it up to questions. There are a few here if there's none from the audience, but I'll open it up to questions from anybody. I'll take some from the Q&A here. So first of all, just from my comment from me is that I just really wanted to commend you on the really exciting work you're doing, and to end on this outstanding work that you're doing on the huge problem of sickle cell anemia. It's really, really exciting. And so some of the questions that have come up is what type of LNPs were used? And I think you just answered that by talking about the support from Acuitas on making the LNPs. So if you want to add anything to that, please go ahead, but otherwise we can just leave it at that.
Yeah, well, we've used a bunch of different LNPs from a variety of companies.
Another question is, how did you functionalize or install the SATA on the antibodies, specifically to selected sites, on the amino acid residues?
Yeah, so for whole IgG, it's a random malamide-mediated conjugation. We're now using SCFV, single-chain antibodies, and there we can label the N, the non-antigen binding part, and do specific conjugations with other chemistries.
Excellent. So the next question, I think you answered this. Actually, you gave some really great examples, and I'll just say, like, I agree that the antibody targeting work is just, it's really outstanding. And one of the questions is, could antibody labeled LNPs be used for targeted delivery in tissues other than liver? And of course, you showed some really good examples. And the question here is talking about lungs, and you showed some at the beginning that we're targeting lungs, but maybe you could. What about, in addition to lungs and bone marrow, are there other tissues that you are interested in?
Yeah, so we've developed targeting to a variety of tissues. I mentioned most of them. We can also target heart, which has a lot of people excited for therapeutics, and we're developing more. So we're ultimately, we like to be able to figure out how to target brain and target specific cells in the brain. That's going to be long-term work. But we're looking at targeting kidneys, targeting mesenteric lymph nodes, targeting GI epithelial cells of a variety of cell types.
Yeah, excellent. And that's great. And we're going to hear about 2 more talks coming up about other tissues of interest that people are working on. And you've just had a lot of comments on just thank you so much again for this fantastic talk. The last one that I'll ask is about targeting again. How well is the biodistribution and gene expression correlated? Do you see big differences? Maybe it's going into one tissue, but it's not expressing as much as you might expect.
So with bone marrow, we have close to 100% of all bone marrow progenitor and stem cells are taking it up. In lung, in brain, we have very high levels of transduction efficiency. So there are off targets. We are targeting the liver. We are targeting the placenta in pregnant females. We are targeting a few other cell types that we'd prefer not to target, but that's a problem of LNP design.
Great. There's a few other questions in the Q&A, but I think, unfortunately, we don't have enough time to do more. But Dr. Weissman, if you have the time, please feel free to answer some of those, but don't feel that you need to either. It's just been really fantastic to have you here. Thank you so much.